Thank you for that introduction. I'm very happy to be here today. My talk is titled, Protecting Participants in Bioequivalence Studies for Abbreviated New Drug Applications During the COVID-19 Public Health Emergency, and it is the same title as a guidance document that the Office of Generic Drugs published in January of this year. That guidance document is also the subject of my talk today. At the conclusion of my talk, you should be able to list the elements of study conduct to be considered when planning to resume or initiate new studies with human participants, describe how study sites should develop or revise procedural documents while taking the risk for COVID-19 into consideration, identify strategies to protect the scientific validity of bioequivalence studies, and explain how study sites should best plan for timely and organized resumption of BE studies. So let's begin. The American public depends on continued access to life-saving medicines, and because 9 out of 10 prescriptions in this country are filled by generic drugs are instrumental in assuring that this access remains uninterrupted. The worldwide COVID-19 pandemic created challenges for all of us, with novel complexities and the ushering in of a new reality. However, throughout the pandemic, FDA's generic drug program remained resolute in its efforts to help increase the availability of safe, effective, high-quality, more affordable drugs in the United States. And part of those efforts is the issuing of guidance documents that provide timely guidance for industry and support response efforts to the pandemic. The title page to the guidance document I'll be discussing is displayed on this slide. It should be noted that the policies herein are intended to remain in effect only for the duration of the public health emergency due to COVID-19. So, FDA recognizes that the COVID-19 public health emergency may impact bioequivalent studies with human participants that are being conducted to support abbreviated new drug applications. And this impact may be due to multiple and various reasons, including limitations on travel and closure of study sites as well as laboratories. As a consequence, these studies may have been suspended at various stages along the way, ranging from protocol development to during or between study periods. So when we're talking about BE studies that involve human participants, the primary objectives are to protect the rights and welfare of the participants and to assure data quality and integrity while at the same time remaining in compliance with all applicable legal requirements. The recommendations in this guidance are intended to facilitate achieving these objectives. And of course, the safety of all involved in these studies, including study site staff, should be taken into consideration. So let's take a closer look at what's in this guidance, beginning with issues surrounding the safety of participants. The agency recognizes that BE studies conducted during the development of generic drugs often involve healthy participants, but also may involve participants in the patient population for which the reference product is indicated. So when considering enrollment criteria, it's important to keep in mind that some members of a targeted study population may be at greater risk than others for severe illness due to COVID-19. And any enrollment strategy should account for differences within a study population, including vulnerabilities such as various comorbidities, as well as the local prevalence of COVID-19. Moving on to visits to the study site. The aim is to minimize exposure to the SARS-CoV-2 virus. And when it comes to the screening consenting process, strategies to minimize exposure may include methods that reduce time at the site and interactions with staff. One method to consider is electronic informed consent, and the FDA guidance from December 2016 can be reviewed for further details. Additionally, input from infectious disease and public health experts may aid in the development of strategies to reduce exposure. Various strategies may be employed to mitigate risk of exposure during the study periods. The guidance provides a couple of examples, but others may be developed and used, and each will have their pros and cons. The first is termed the confinement or bubble design. Here, participants involved in the study and potentially site staff are confined to the study site for the duration of the study. Regular screening for COVID-19 symptoms would be performed, as well as other relevant practices such as personal protective equipment use, or PPE, frequent hand washing, and bedside sampling. The second design described in the guidance is termed the non-confinement or ambulatory design. In this model, there would not be any overnight stays at the facility, and select time points would be chosen for pharmacokinetic sampling that would allow participants to travel to and from the facility on a daily basis. This model would be expected to help minimize participants' exposure to facility personnel and other participants. COVID symptom screening would be performed at each check-in. With regard to sampling, the guidance mentions a few approaches that may help to mitigate exposure risk. The first is using an alternative PK approach that would optimize sampling time points that minimize ambulatory visits. Where applicable, use of modeling or PK parameters such as partial AUC could be investigated, and the acceptability of these alternative approaches may be considered by FDA provided that applicants provide justification that their proposed approach does not compromise the accuracy, reproducibility, and sensitivity of the BE test. Another approach to reduce exposure between participants is to use spaced dosing and sampling, such that visits do not overlap. Staggering the initial dosing would ensure subsequent visits would be similarly spaced. This method has the potential to increase study duration, and it's important to note that statistical considerations to reduce bias should be pre-specified and clearly described within the statistical analysis plan. Also, off-site or at-home sampling may be an option. There are various ways this can be carried out. Examples include participants collecting samples and mailing them, or study personnel picking them up, or even having study personnel coming to the home for sample collection. As is generally the case, we recommend that validation for an alternative method follow the agency's Guidance for Industry Bioanalytical Method Validation published May 2018, and that a correlation study to assure that samples collected at home would provide the same results as whole blood samples taken at the site should be performed. On the back end, follow-up strategies to reduce in-person follow-up may include performing end-of-treatment procedures at the time of discharge and conducting subsequent follow-up visits virtually. Of course, this strategy would not preclude the need for in-person examination and follow-up for those participants who have experienced an adverse event. This slide is a reminder that detailed plans or SOPs should be developed to help delineate and disseminate mitigation strategies and ensure study integrity. Consultation with local and federal guidelines for infection control in business and healthcare setting is recommended. Protecting scientific validity. This section of the guidance is provided to help applicants protect participants' rights and welfare and assure data quality and integrity while maintaining compliance with applicable legal requirements. This first slide deals with protocol amendment and development. Typically, changes in protocols are not implemented prior to review and approval by the Institutional Review Board or Independent Ethics Committee, and for those BE studies conducted under IND by FDA. However, when urgent or emergent changes to a protocol or an informed consent are necessary to eliminate immediate hazards to study participants, they may be implemented prior to IRB or IEC approval and filing of an IND amendment. In these cases, the applicable parties are encouraged to engage with their IRB or IEC and, as applicable, FDA. These changes must be reported afterward and should be reported in the ANDA with sufficient information and justification. Protocols should pre-specify how participant illness will be handled, and protocol and statistical analysis plan changes should be made prior to data lock and unblinding. For example, describing the handling of missing data along with justification of the chosen method. With regard to protocol deviations, the COVID-19 public health emergency has generated unavoidable deviations, and good documentation for these deviations should be maintained, as well as maintaining an open line of communication among study sponsors, monitors, investigators, and IRBs or IECs. Study oversight and monitoring have been impacted, and unique challenges have been created. Considerations for remote monitoring may be an option in some circumstances. For more in-depth information on this topic, I refer you to the Guidance for Industry Investigators and Institutional Review Boards titled FDA Guidance on Conduct of Clinical Trials of Medical Products During COVID-19 Public Health Emergency, published March 2020 and updated September 2020, as well as the Guidance for Industry titled Oversight of Clinical Investigations, a Risk-Based Approach to Monitoring, published August 2013. With regard to drug expirations, in general, we do not recommend the use of expired reference or test product that is out of specification due to aging. The Guidance for Industry titled Development of Abbreviated New Drug Applications During the COVID-19 Pandemic, Questions and Answers, published April 2021 and updated September 8, 2021, addresses some of the questions surrounding this subject, and others may be posed through controlled correspondence. Lastly, when planning for resumption of studies, sites should comply with applicable public health guidelines and requirements and consider the safety of both participants and staff. And it is our intent that this guidance document be used as an aid to help sites be better prepared to either resume or begin the conduct of BE studies with human participants during the COVID-19 public health emergency. Okay, so on to the challenge questions. Question 1 reads, which of the following are examples of COVID-19 infection mitigation models? Choices are A, the confinement or bubble design, B, the containment or sanitization design, C, the non-confinement or ambulatory design, D is A and C, or E is all of the above. Take a few moments to think about your choice of the correct answer. Okay, so if you chose D, A, and C, you're correct. If you recall, the confinement or bubble design is a design where subjects and potentially study site staff remain at the site for the duration of the study. And C, the non-confinement or ambulatory design, is a model where participants come to and from the study site on a daily basis and are screened for COVID-19 symptoms at each re-entry. The containment or sanitization design was not mentioned in the guidance document. Okay, question number 2 reads, protocol revisions or modifications made while conducting a study during the COVID-19 pandemic do not need to be reported to the IRB or IEC. Choices are A, true, and B, false. So again, take a few moments to think about the correct answer. Okay, if you selected B, false, then you are correct. And so if you recall, when urgent or emergent changes to a protocol or an informed consent are necessary to eliminate immediate hazards to study participants, they may be implemented prior to IRB or IRC approval and filing of an IND amendment if that particular study is under IND. However, these changes must be reported afterward and also should be reported in the ANDA with sufficient information and justification. These next 2 slides provide references for my talk and also hyperlinks to the relevant websites and guidance documents. I'd like to leave you with this closing thought. Adherence to the principles of good clinical practice, including human subject protection, is universally recognized as a critical requirement to the ethical conduct of research involving human subjects. I thank you for your attention today.
Thank you for the kind introduction. Today, I'd like to talk alternative bioequivalence approaches for data analysis due to COVID-19 related study interruptions. This presentation represents the views and perspectives of the speaker and does not necessarily reflect the views of the US FDA. From this talk, I will describe common challenges for conducting bioequivalence studies during the COVID-19 pandemic and how OGD can help prospective applicants for their COVID-19 interrupted studies. I will also bring a case demonstration to explain the use of alternative data analysis approaches for a case of a COVID-19 interrupted in vivo BE studies. The COVID-19 public health emergency has presented challenges to drug development, including the BE studies that intended for submission in ANDAs. Planned BE studies may be interrupted by different ways, such as exposure risk, travel limitations, site closures, and product availability. The interrupting and restarting BE studies may make it difficult to meet some protocol-specified procedures for clinical study exclusion, also impact the information needed to establish bioequivalence. OGD has published guidance regarding the conduct of in vivo BE studies during the COVID-19. The OGD's approach is to bring everyone to the table and brings multiple disciplines to provide consistent, timely, and scientifically sound advice. The COVID-19 pandemic may bring some novel and unique challenges to generic drug development. The prospective applicants can submit specific questions related to their impacted BE studies via the control corresponds process or other process. The questions may include, but not limited to, the question for the proposed protocol modifications, such as alternative statistical analysis plans as supported by modeling and simulation, or modifications to incorporate adaptive designs that may allow recruitment of additional subjects. FDA encourages prospective applicants to find and perform alternative analysis approaches for COVID-19 interrupted studies. Per guidance published for study conduction during the COVID-19, any protocol or statistical plan changes should be accompanied with adequate justifications and not lead to biased equivalence determination. In addition, protocol and statistical analysis plan changes should be made prior to data lock and unblinding. Here, I summarized some common questions on BE study due to the COVID-19. First, their test and reference availability inquiries. For example, the product may expire because of the study interruption. In general, only a single manufacturing batch of an R&D is used for a single BE in vivo study. However, if a product expires, multiple batches of a product may be used in a single PK study, or different batches may be used between fed and fasting study. The second very common inquiries are protocol revision questions, such as interim analysis or adaptive design, and shortening study durations or truncated approach, and change of study designs, such as changing crossover design to a parallel design. There are other inquiries too. Because of the COVID-19 interruptions, there might be study with large missing data or study conducted partially, such as conduct the fasting study only but not the fat study, if two studies are required, or use in vitro only approach, or to conduct remote study. In today's talk, I would like to present a case demonstration that used quantitative method and modeling approach to assess COVID-19 interrupted in vivo PKBE study with 2 reference batches. This case question came from one of the common questions I just showed, which is a product expiration problem. In general, we do not recommend use of expired product. So, when a reference product expired in an ongoing BE study due to COVID-19 related interruptions, for a study that is interrupted in the middle of the study, there can be the case that use one batch of the product in one period and use a different batch of the product in another period for a crossover design. So this case demonstration is to address a question here. If 2 R batches are used in a pivotal PKBE study, how can we assess the BE result? We used a replicated 4-way crossover design as an example for this case demonstration and used a hypothetical NTI drug as an example. As recommended in the guidance, statistical approach to establish BE, the same lots of the test and reference product should be used for the replicated administration. This can avoid introduce of the batch-to-batch variations. For the NTI drug, clinical efficacy and safety are sensitive to the systemic drug exposure. Given this nature, the BE limit is scaled to a narrow range based on the within-subject variabilities of the reference product, as shown in the figure on the left. The blue and the red lines are showing the implied BE limit on TR ratios that was scaled by reference drugs within subject variability and can be kept at 80 to 125% as a maximum. The table on the right shows the corresponding BE limits to different within subject variabilities. Our simulation was performed with a hypothetical orally administered product with 2-compartment model using population PK approach. The investigated scenarios include 2 types of interruptions. As shown in the table here, the first one is an interruption happens after the completion of period 3. So in this case, period 1, 2, and 3 are conducted with the first batch of the R&D, R1, and period 4 is conducted with the second batch of the R&D, R2. Our current study only considered the reference drug's expiration, so all 4 periods will be using the same batch of the test product, shown as T here. Another interruption type we simulated is to interrupt after the completion of period 2. So period 1 and 2 are conducted with R1, and period 3 and 4 are conducted with R2. For comparison, we also simulated uninterrupted studies, which are completed with one batch of the product. So we simulate one with R1 only, and another one with R2 only. In the simulation, we simulated a range of underlying R batch differences by changing the relative bioavailability factor to create R1, which is batch 1 of the reference product, and R2 for the batch 2 of the reference product. We also want to create different scenarios of the TR ratios. Simulations were conducted for a wide range of the geometric means of TR ratios. It's like a scanning process. So it covers case that T is on the left side of R1 and R2, and T is at the middle of R1 and R2, and T is on the right side of R1 and R2. Also, simulations are able to cover bioequivalent cases, the borderline bioinequivalent cases, and clear bioinequivalent cases. Our assessment is based on comparing BE outcomes for the interrupted cases that conducted with 2 R batches versus the uninterrupted cases. When a study passed the interrupted design, we use the BE results from uninterrupted study to represent the possible outcomes if there were no study interruptions, to decide if we can accept the BE result. When compared with uninterrupted studies, it can be different scenarios. The first one, the T can pass BE for both T versus R1 and T versus R2. In this case, we consider the result is acceptable. The second one, for T can pass BE for either T versus R1 or T versus R2, but not for both, which is acceptable too. The third case, T cannot pass BE for either T versus R1 or T versus R2, and PK exposure of the T falls within the PK exposure of the 2R batches, which is acceptable too. But for the last case, that T cannot pass BE for either T versus R1 or T versus R2, and the PK exposure of T falls outside those of the 2R batches, we consider this is a failure scenario for not being BE. In other words, if a test passed BE in interrupted studies that conducted with 2 batches of R, we compare this past result when it conducted without interruption, and we look to see if the study falls into this 4 which category to inform if the BE results is acceptable. This is our analysis scheme. First, we conduct a preliminary ANOVA test to see the potential batch effect by using batch nested with treatment as a fixed effect in the statistical model. For study with no significant batch effect, that p-value is larger than 0.1, we conducted a conventional reference scale BE study approach without considering the batch effect that exclude the batch from the statistical model. And we collect all the past study to assess the BE outcome with the uninterrupted studies to assess if this BE outcome is acceptable or not. For the study with significant batch effect, for the p-value is smaller than 0.1, we will conduct an alternative reference scale BE that included batch as a fixed effect in the statistical analysis. Also, we collect the past study and compare it with the uninterrupted studies. Here is the preliminary ANOVA test results. Although we do not expect batch-to-batch difference to be as high as 30%, but we conducted batch variations from 5% to 30% for illustration purpose. From the table, we found that the percentage of study with significant batch effect increases with increasing batch-to-batch variation. When batch-to-batch variation is higher or equal to 20%, more than 95% of interrupted studies showed significant batch effect regardless of the types of the interruption. This is the analysis for studies that evaluated with conventional reference scale BE approach, with the batch effect excluded in the statistical model. The table indicates the outcome distribution in terms of the percentage for these 4 scenarios when compared with uninterrupted study design. Here, the 1, 2, 3, 4 is the 4 categories that I previously showed when compared with the uninterrupted studies. We would like to see the percentage of study falls into the failure scenarios, which was highlighted in the red in this table. The top half of the table shows the interrupted studies with 3-to-1 division for the R batches, and the bottom half shows the study with 2-to-2 divisions. We can notice that the chance for BE outcomes to fall into the BE failure scenario is close or equal to 0, across all the studied scenarios. Now let's take a look at the interrupted study that shows significant batch effect that evaluated with alternative approach by incorporating batch as a fixed effect. Again, by comparing the uninterrupted study, the B outcome would fall into 4 scenarios as indicated in the table. We can see that most of the cases, the result is acceptable by comparing with uninterrupted studies. The chance of study fall into the BE failure scenario, however, it's more than 5%, as highlighted in red here, when the batch-to-batch variation is small, like 5%, and interrupted by a 3-to-1 R batch division. The chance of falling into the BE failure scenario was not observed for studies with 2-to-2 R batch divisions. This is the conclusion from the case demonstration. From simulation results, the B results obtained from interrupted studies with no significant batch effect seem to be acceptable to use a conventional statistical analysis approach with batch treatment terms excluded. However, the acceptability of B outcomes from interrupted studies which has significant batch effect using the alternative statistical approach with the batch term included, the result may be case-specific. In conclusion, the simulated scenarios are only considered as case demonstration, which cannot be extrapolated to all interrupted studies. The study result could be case-specific. Overall, FDA is proactively evaluating approaches to mitigate study challenges posed by the COVID-19 pandemic. Simulation can be one of the approaches to show a modified BE method is acceptable. For any protocol or statistical analysis plan changes, it should come accompanied with adequate justification and not lead to biased equivalence determination. Industry can include science-based justification for alternative approaches to data analysis from interrupted studies, and they should pre-specify analysis plan before analyzing the data. The proposed framework can be discussed with FDA via the control corresponse or other appropriate method. Question 1. Which of the following statement is not a common question on BE study due to COVID-19? A. Product expiration, usage of multiple batches of the product in a single PK study. B. Study with large missing data. C. Subject dropout due to adverse events. D. Shortening study duration, truncated approach. Take a moment to consider which answer you think is correct. For those who pick answer C, you are right. Question 2. For COVID-19 related study interruptions. A. FDA encourages prospective applicants to find and perform alternative analysis approach for COVID interrupted studies. B. Any protocol or statistical plan changes should be accompanied with adequate justifications and not lead to biased equivalence determination. C. Protocol and statistical plan changes should be made prior to data lock and unblinding. D. All of the above. The correct answer is D. All of the above. For more information, please find on the FDA website. I would like to acknowledge all offices that support this effort, especially to the following individuals. Thank you. I'll be back on the panel discussion. Thanks.
Thank you for that kind introduction. Hello, everyone. My name is Gloria Huang with the Office of Life Cycle Drug Product in OPQ CDER FDA. I'm here to present FDA guidance, temporary policy on repackaging or combining Propofol drug products during the COVID-19 public health emergency. Drug product quality consideration. The objective of this presentation is to recognize the purpose of the guidance, review the Propofol drug product and their physical-chemical stability properties, discuss the repackaging or combining Propofol drug products based on the compatibility. This guidance has been published in April 2020. The link to the guidance is provided here for your convenience. The guidance is issued to address the COVID-19 public health emergency. Why this guidance is needed? First, Propofol is on the drug shortage list. Hospitals have had difficulties in obtaining adequate supplies to care for critically ill COVID-19 patients. Healthcare professions request guidance on repackaging or combining smaller size of drug product into larger vials. For example, 20 ml to 50 or 100 ml stereo container to supply shortfalls of the larger size product. FDA-approved label states Propofol undergoes oxidative degradation in the presence of oxygen. It is therefore packaged under nitrogen. That means exposing Propofol to oxygen during repackaging would be in conflict with the labeling and would fall outside the policy in the repackaging guidance. Pharmacists asked about combining the content of different manufacturers' Propofol product and placed them in the same container. Propofol is not covered by other temporary policies for compounding of certain drugs for hospitalized patients during COVID public health emergency because of the quality concerns. The purpose of the guidance is to provide temporary policy regarding the packaging or combining Propofol by state licensed pharmacy, federal facilities, outsourcing facilities for use in the hospital that are having difficulty obtaining adequate supplies. To address the certain practice falls outside the FDA repackaging guidance. This guidance is valid only during the period of COVID-19 public health emergency or until withdrawal by FDA as no longer appropriate. Propofol drug products is an IV general anesthetic and sedation drug. The approved label for indication and usage include multiple indications. For different patient populations, the age can be 2 months to adults. This slide summarizes the FDA-approved drugs per Orange Book. As you can see from the table, that the first Propofol product approved by FDA in 1989 as Diplodin in NDA 019627. Since then, several drug products have been approved, and Diplodin is now the reference listed drug. Propofol is slightly soluble in water. The pKa is 11. The partition coefficient at the pH range 6 to 8.5 is 676121. The drug product is formulated in a white oil in water emulsion, consists of Propofol 10 mg per ml, soybean, glycerin, egg lecithin, and preservatives, with sodium hydroxide to adjust pH. Preservatives may be different for different products by different manufacturer. This slide shows the packaging sizes of reference-listed drug Diplovan NDA 019627. The chemical structure of Propofol is given on the right side. In the whole supply section for U.S. products, the NDA Diplovan supplied in U.S. as package size 20 ml, 50 ml, 100 ml. Also, the label indicated that Propofol is oxidative in the presence of oxygen and should be packaged under nitrogen. And the product should also be stored between 4 to 25 degrees without freezing. Shake well before use. The label also states that strict aseptic technique must always be maintained during handling. Propofol injection emulsion is a single-access plunger product, meaning for single patient infusion vial, which contains preservatives to inhibit the rate of growth of microorganisms for up to 12 hours. Propofol drug products can still support the growth of microorganisms, as it's not an antimicrobially preserved product under USP standards. Do not use if contamination is suspected. Discard unused drug products as directed within the required time limits. There have been reports that mishandling Propofol drug product may cause microbial contamination of the product and may cause fever, illness, infection, and death. It's also reported in literature that misuse of Propofol drug product can cause transmission of bloodborne pathogens such as hepatitis B, C, and HIV. Specifically, use Propofol vials intended for single use on multiple persons. The product has to be checked well before use. Do not use it if you see excess creaming or aggregation, if large droplets are visible, or the face separation. Slight creaming may be visible upon prolonged standing, but it should be disappeared after shaking. This slide shows an example of vial and carton label for generic product. Preservatives are provided on the label of vial and cartons as pointed by arrows. The preservatives used in currently approved drugs are EDTA, sodium metal bisulfite, benzyl alcohol combined with sodium benzoate, benzyl alcohol, and sodium benzoate. The last three preservatives are very similar. If combined, the risk is low. Improper packaging or combining may cause characteristic changes such as physical chemical properties change, and may negatively affect the safety and effectiveness of the drug product. For example, impurity level increases due to the degradation of drug product. Repackaging the stereo drug product may cause microbial contamination due to mishandling. This could cause patient injury and death. The physical, chemical, and stability properties for drug products considered for repackaging or combining include appearance, pH, type of preservatives, container closure system, and packaging sizes, stability data, expiration data, in-use study. Additional attributes such as mean globular size, p-fat5 are also considered because they are important for this product. Based on EP quality information, the enforcement disclosure policy has been developed. As a template measure, in the following circumstances, FDA does not intend to take action for certain violations regarding Propofol drug products that are within expiry and in their original containers. The repackaged or combined drug products is provided directly to hospital that informs the pharmacy or outsourcing facility that the drug product is treating patients with COVID and has made reasonable attempts to obtain adequate supplies but has not been able to do so, as described in section 3.1 for the guidance. In all cases, the repackaged or combined product is discarded if there is any changes in appearance such as color, visible separation of water and oil occurs, such as formation of any visible oil droplets or particulate matters. See section 3.2 in the guidance. The product is repackaged or combined consistent with the practice described in the repackaging guidance, except that with respect to combining products, the application of but product labeling, and container closure. See section 3.3 in the guidance. So FDA approved Propofol products may be repackaged or combined without doing so under nitrogen if Propofol mixed using aseptic technique under ISO 5 conditions whenever possible. This will provide flexibility during the public health emergency with limited time period preceding use of the packaged product. When the FDA approved drug product containing the same preservatives placed into a new container, beyond the use date of not more than 12 hours is used, and the resulting product is not frozen. See section 3.3b in the guidance. This means different lots of Propofol from the same manufacturer may be mixed under certain circumstances. If there is a difference in preservative or antioxidant, the two products are not compared, except as follows. See section 3.3c. ANDA 077908, 205067, 205307 contain very similar preservatives. Combine them is a low risk and the public health emergency. Mix the three products will result in a final mixture with slightly less benzyl alcohol, so a batch use data of not more than 4 hours can be used as a precaution against the microbial growth in the presence of reduced preservative content. Lastly, Propofol product under AND 019627 and AND 075102 are not combined with any other Propofol product because there are significant differences in the preservatives. The container used for repackaging or combining is suitable for storage of drug product through its butt. See section 3.3d in the guidance. Because the Propofol may absorb into the plastic or rubber stop packaging components, which may impact efficacy. The labeling of repackaged or combined drug product specifies the storage condition between 4 to 25 Celsius degree and do not freeze. The tubing or any unused portion of Propofol drug product should be discarded after date and time corresponding to bud. The section 3.4 in the guidance for pharmacy facility and labeling is not covered in this presentation. The enforcement of discretion policy I just discussed is summarized in Appendix X in guidance. The table includes 5 columns, each represents one manufacturer's product. And in each column listed the distributors and packagers. Next page is more packagers. The repackaging or combining should be consistent with the policy in the guidance. Here are 3 main points. Products included in the same column may be mixed with bud date not more than 12 hours. This is for the same preservatives. Propofol products in column A and B are not mixed with any other columns other than its own. This is significantly different preservatives. Propofol products in column C, D, E may be combined with the bud date not more than 4 because of the similar preservatives. Now let's look at some challenge questions. Question 1. What are the FDA-approved indications for Propofol drug products? A. Antibacterial. B. Antivirus. C. Anti-inflammatory. D. Anesthesia and sedation. Let's take a moment to think about what's correct. If you choose D, that's correct. Challenge question number 2. Up to how many hours are the preservatives in Propofol injectable emulsion expected to inhibit the rate of growth of microorganisms in the event of accidental intrinsic contamination? A. 4 hours. B. 10 hours. C. 12 hours. D. 24 hours. Take a moment to think about what's correct. If you choose C, that's correct. Congratulations. I have covered a lot of materials. In summary, we have discussed the temporary policy of repackaging and combining as described in the guidance to address COVID-19 public health emergency. We discussed the quality information including labeling and storage conditions, formulations, physical-chemical properties, stability data of approved products. I would like to thank my colleagues in Office of Life Cycle Drug Product and Office of Policy for Pharmaceutical Quality for their contributions to the guidance, for their help in preparing these presentations. Thank you. I will be back to answer questions on the panel.
Hello, my name is Kaijun Feng. People call me Kevin. I'm from Office of Research and Standards from OGG CDER. Today my topic is applications and lessons learned for conducting adaptive designs in generic drug development. So before I start my presentation, here is this disclaimer. This presentation reflects the views of the author and should not be construed to present FDA's views or policies. So the learning objectives of this presentation is to list some of applications and lessons learned for conducting adaptive design in generic drug development. The second objective is to clarify the acceptability of adaptive design for the submission in abbreviated new drug application that's entered during the COVID-19 pandemic. So two objectives here. So by looking at the adaptive design, look at the history and definition. The history is started from 1970s. It defines as a group sequential. And then in 1990s, they call it blinded sample size risk estimation. And more recently, there are a few of new definitions. For example, unbounded sample size risk estimation, adaptation to patient population, adaptation to treatment arm selection, and patient allocation, other aspects of study design. And then in the FDA guidance, it defines a clinical trial design that allows for the prospectively planned modification to one or more aspects of the design based on accumulating data for the subject in the trial. So prospectively planned and accumulating data from subjects in the trial. So here I give you some of the application of adaptive design, and the features including the possibility of stopping a clinical study earlier based on interim study results, or adjusting the sample size during the study. Here a list of the ENDA submission from 2006 to 2019. There are three categories. The first one is a two-stage design. Second one is a group sequential design. The third one is futility interim analysis. By looking at the number of ENDA submissions, two-stage design have 9, group sequential has 3, but futility interim analysis has 34. By looking at the area in the product, two stages including oral injection and topical, and the group sequential also including the oral and topical. However, the futility interim analysis only including topical as product. For the feature differences, the two-stage design is to risk estimate the sample size based on the stage 1 non-BE study. And then for group sequential, it predetermines sample size and tau and error at each stage. So the difference between these two is for two-stage design is risk estimate sample size. For group sequential design is to predetermine sample size. But for futility interim analysis, it's really to stop only if deemed not BE. I'll give you some case example to explain this further. So first, a case example. So in this case, a randomized two-stage using the Propofol method C and the single dose and crossover PK study with AUC 0 to T and Cmax PK endpoints. So for this design, here is the graph that we explain how the Propofol C method, and I explain what is Propofol C, and there's a two-stage of design. The first stage is on the top, and second stage was on the bottom here. On the first stage, that using the alpha level is 0.05, if the power study, they do the power analysis first, evaluate greater than 80, then do the BE at stage 1. If it passed, then it passed. If it failed, then it failed. And if the power is less than 80, and then using change the alpha level, the significant level to 0.294. So if BE is met, then you can stop, and then you will pass. However, if it's not, BE is not met, and then can calculate the sample size based on the stage 1's analysis using alpha level 0.294, and it continue to the 2-stage. So there's a 2-stage design, and then using both of the stage using alpha level 0.294, and then stop the analysis. If it's passed, then it will pass. If it's not, then it will fail. So for this particular case, study 1, the applicant showed that the is passed because the power already greater than 80, and it passed. So from this design, you can see that the applicant start this adaptive design earlier, only at stage 1 without going to stage 2. That probably saves some time and the sample size and the cost as well. For the study 2, this is a pivotal B study for topical products on the PD endpoint, AUEC evaluation. So the interim analysis at 60% of subjects. So that means there's 49 subjects out of 80 total planned subjects was used for analysis. The results show that the BE not deemed to fail. Therefore, the will continue until the protocol specifies 80 subjects, and then eventually the 90% confidence interval of the TR ratio of the PD endpoint fell within the 80 to 125. This demonstrates a successful BE study. So on the other hand, if the 60% of subjects is deemed to fail, then they can stop the study early. So that means they can stop the risk earlier before the full subject was conducted. So here is a case 2 study. However, we also observe some of the submission, and that the lesson we can learn why the adaptive design failed at the very beginning. So the first one, for the submission, did not pre-specify a study and adaptive design for the stopping or continuous study, so not pre-specified. The second one did not follow the procedures and statistical analysis plan pre-specified in the protocol. For example, the study power analysis was not including before the B evaluation at stage 1. So the statistical analysis specified in the protocol, but the power analysis was not included, and did not define additional subject as a post-hoc add-on study in sequential or group sequential design, but was included in the data analysis. So this is a post-hoc add-on, but not pre-specified. So these 3 lessons that have been learned and has not been successful for the BE adaptive design at the very beginning. And for the adaptive design, about the time and error, and I would like to give you additional information on the 2-stage adaptive design using the POP-in method. So the agency will accept appropriate design bioequivalence study if the applicant provide evidence that your proposed approach is validated. And then the approach does not inflate time and error can be spotted by either peer-reviewed journal papers with the same study design and by simulation method. So the 2 types that you can support. One is peer-reviewed journal that not inflate time and error. The second one using simulation method, for example, the time and error control at the nominal level with 0.05 for bioequivalence test. So here I come with challenge question 1. So one of the following statement is not true. So the first question is, when BE fails and the power less than 80% at stage 1 of the two-stage design using pop-in-C method, the applicant can continue to stage 2. Second question is, at futility interim analysis, the applicant can stop the study when BE was deemed to be unsuccessful using 60% of the planned subjects. I'll give you a couple seconds to think. C. Additional subjects was included for data analysis when the subjects was pre-specified in a group sequential design. So here D. When BE passed and was evaluated using alpha equal 0.05 at stage 1 without power analysis, BE is concluded as a successful study. So which one is not true? So you can make your decision. So here are the results that the D is not true. When the BE passed, evaluate using alpha 0.05 at stage 1 without power analysis. So the power analysis should be done before this be evaluated. So this is the one of the mistakes that was taken by the one of the submissions. And the second one, I would like to focus on the adaptive design during the COVID-19 pandemic. So here is some of the reference that you can refer to. So one of the reference was published by for the ENDA submission during the COVID-19 pandemic, is to provide detailed information including proposed plans, rationale, and justification supporting your proposal when communicating with the agency. The second one is what are the challenges and potential opportunities for implement adaptive design for BE study on generic drug development. So here is one of our publications that you can refer to what is the challenge and what is the lesson that you can learn from this study. So here I give you the third case study. So in this study, the ENDA submission for a fully replicate BE study for the fasting and fat. And the product specification is available for this product. And also some of the ENDA was approved. So that means this should be an easy product for the applicant to conduct and to be a successful BE study. However, the COVID-19 comes and then about 20 out of 60 recruit subjects was not included. That means 1/3 of subjects was not included because of the COVID-19 situation. So those missing data because of at least one period were missing. So that's 40 subjects was used analysis. So here I give you some of the challenge question, and then you can think of what can you do with this situation. So the one of the comments is not true due to the COVID-19. So first one, A, the applicant can do the BE data analysis based on the available subjects. If it fails, additional subjects can be added to increase the power as an adaptive design. The second one, B, the applicant can do the BE data analysis based on the available subjects. If it passes, BE is concluded as a successful study. C, the applicant can recruit additional subjects by pre-specifying as an adaptive design in the protocol applied to data unblending. D, the applicant can send a controlled correspondence and ask the agency's agreement for recruiting more subjects as an adaptive design prior to data log. So you can think about it and make a selection for one of them is not true during the COVID-19. So here is the one is not true, because when it failed, additional subject is added at the end. So the rest of them is true. I will give you some of the example why it's not true and what is the guidance will tell us, because it's already failed and they add on the additional subject to increase power for the study. So here come the results of the case study. So give you results. So the result is because the sample size will reduce, 1/3 will reduce, and then it results in wider confidence interval and failed the Cmax in both studies for the reference scale average criteria. So the study basically failed on the reference scale average on Cmax. And without the interruption by COVID-19, Cmax for the reference scale average bioequivalence criteria for both studies could have been met and therefore could be concluded. However, because the COVID-19, a lot of data were missing. So this is the one of the COVID-19 affected design. And in the guidance for the COVID-19 statistical consideration for clinical trial during COVID-19 on the public health emergency published on June 19, 2020, is specified that for the analysis or updated statistical analysis plan before the locking the database. So before locking the database. And the second guidance for the COVID-19 by the FDA guidance of industry for protecting participants on BE study for end COVID-19 public health emergency is still specified. So those change and statistical analysis should be pre-specified in your statistical analysis plan with sufficient information and justification, and may apply to data lock and unbundling. So in terms of post-hoc analysis, so how can we define the post-hoc analysis? So any additional analysis after data lock and unbundling are post-hoc in nature. The agency considers post-hoc analysis as hypothesis generating and generally expect a prospectively designed study to be used to establish BE before and end submission. The study protocol should specify how missing data will be handled, and applicants should justify the method chosen, and including the modeling simulations. So you can use in the modeling simulation to pre-specify the study protocol, how you handle the missing data. So that we can help you for your pre-specify your design. For example, the adaptive design you want to add. And here is the power study, because the sample size will reduce. And then it is the applicant's responsibility to design and adaptive design power B study for the proposed study design, and the acceptability of your proposed adaptive design will be determined during the scientific assessment of end submission. So those, because of COVID-19, there's 1/3 subject affected. How can you add on for those situations? So you have to think, and the guidance available for you to act on this specific case. From this lesson, we can learn the protocol should prospectively identify how the study will handle participant illness, either from COVID-19 or some other reason during the conduct of a study. So, for example, to consider whether to remove, replace, or provide another method to address missing data for that participant. Protocol and study statistical analysis plan change should be made prior to data lock and unbundling. Prior to data lock and unbundling. For example, the protocol should be prospectively states how missing data will be handled in statistical analysis and provide appropriate justification for the method chosen. So here we have the summary for the adaptive design. So those applications and lessons learned is really to help future adaptive design in generic drug development, so that you know that what can be successful, what is not successful, and what kind of thing that you need to do it properly, including to read the guideline carefully and act before those that happen. The second one, you can always communicate with agency, and that communication is really encouraging. And for the adaptive design during the COVID-19 pandemic, we apply to that a lot and unbundling. And here are some of the reference that list that you can use. Some of its guidance, and we also have a one publication that you can refer to by John Lee and myself and some other colleagues. And some of the guidance that you can be used and refer to how those adaptive design can be referred to. So here concludes my presentation. We will start our panel study shortly and answer your questions accordingly. Thank you.
Thank you to all of our presenters for their great presentations. We'll now enter into our first Q&A panel, comprising of our session speakers and introducing Dr. Kimberly Witzman, who's a physician and the Deputy Director for the newly created Office of Safety and Clinical Evaluation within OGD. As the Deputy Director, she's responsible for concentrating on complex, long-range, and emergency problems as issues in the area of clinical evaluation of generic drug products and ANDAs, and she serves as the authority on clinical review issues and complex combination products. Also, we welcome Dr. Stella Grosser, who's Director, Division of Biometrics 8 in the Office of Biostatistics in CDER. This division provides statistical support to the Office of Generic Drugs. She's been with FDA for 21 years, beginning as statistical reviewer for new drug products and serving as a team leader before assuming her current position. Looks like we have a few questions coming in right now, and our first group of questions will be directed to Dr. Frost. And here's the first question for Dr. Frost. Can you please clarify if any alternate designs can be adopted for NTI drugs with long washout periods during the pandemic?
Thank you, Ray. By alternate designs, I'm assuming the question is targeted towards the COVID-19 infection mitigation models that I went over during my talk. And I just would like to emphasize that the examples in the guidance are in no way exhaustive. Again, the goal is to minimize risks of exposure for participants and staff. So when a model or design is being developed and chosen, multiple factors come into play, including the drug product, study design, site resources, and of course the local prevalence of COVID. Therefore, a one-size-fits-all approach is really not possible, and sites may choose one of the examples in the guidance, a hybrid of the two, or a customized design that fits their needs after consultation with infectious disease experts and public health experts.
Thank you for responding to that question. We have another question for Dr. Frost, and here's the question. Is a COVID-19 test like antigen testing or RT-PCR a must before check-in?
So again, with these unique challenges from COVID and sort of this new reality, really one-size-fits-all approach is not is not possible. So I think that the COVID tests, the types of tests, and other methods that are employed to screen study participants for COVID infection may vary with local prevalence and with study design. So I guess, for guidance for any specific site, I would refer them to the CDC site on on interim infection prevention and control recommendations for healthcare personnel during the COVID-19 pandemic. And a link can be found to that site on page 4 of the guidance document that I reviewed during my talk.
Thank you for responding to that question. We have one more question for Dr. Frost, and here's the question. Does the FDA require BE studies in healthy volunteers to be monitored by sponsors?
Great, thank you. So for sponsor requirements for monitoring and reporting safety data, I would refer them to the guidance for industry and investigators. It's titled Safety Reporting Requirements for INDs and BE studies, and it was published in December of 2012. And it's intended to help sponsors and investigators comply with the safety reporting requirements.
Thank you for responding to that question. The next few questions will be directed to Dr. Gong. And here's the first question. Is the same R used for in vivo BE studies also used for in vitro BE studies?
Usually the R used for in vivo study should be the one of the batches that are tested in the in vitro studies. So the case demo here is for the case that our product expired in an ongoing in vivo study because of the COVID-19 interruption. And usually we don't recommend use of expired product to conduct the BE study. So you can refer to the FDA guidance for conducting the BE study for ANDAs during the COVID-19 public health emergency and submit the questions related to your expiring product to FDA.
Thank you for responding to that question. The next question for Dr. Gong is the following. If for PBE studies 3 batches of RLD are required and the same is not available in the market, can we conduct with 1 or 2 batches of RLD, and during ANDA review process, can we submit another batch data?
For modifications of study protocols, so the best way is to submit that specific question to FDA. You can view control corresponds and try to provide detailed information, including your proposed plans, rationale, and justification supporting your proposal when you communicate with FDA. Thank you.
Thank you for responding to that question. We have one more question for Dr. Gong, and here's the question. For COVID-interrupted ANDAs, when comparing T to 2R batches, what alpha level should be used?
So here, the alpha level we used here is 0.05. So the use of 2R batches in the case demonstration here is not an adaptive design, and we used regular alpha level. Thanks.
Thank you for responding to that question. The next group of questions will be directed to Dr. Huang. And here's the first question. What are the major formulation differences between Propofol drug products? Also, what are the typical preservatives for Propofol drug products? What are considered as similar preservatives? And also, when can the Propofol drug products be combined, and when can the Propofol drug products not be combined?
Thank you for the question. The major difference in the formulation for Propofol drug products are preservatives. The typical preservatives used are EDTA, sodium metal bisulfite, benzyl alcohol, and sodium benzoate, or combination of benzyl alcohol and sodium benzoate. So this benzoate, sodium, and benzyl alcohol, with combination, they are considered as similar preservatives. The product with the similar preservatives can be combined with shorter beyond use date per this guidance, and product with very different preservatives, such as sodium metal bisulfate, cannot be combined with any other product. Thank you.
Thank you for responding to that question. The next question is also for Dr. Huang, and here's the question. What are the regulatory requirements for relabelers and repackagers?
Thank you for the question. My presentation covered the quality consideration for this guidance and did not include the information section on the guidance for pharmacy facilities or labeling. I would refer you to the agency's repackaging guidance for general requirements for pharmacy, labeling, and facilities. For specific requirements regarding Propofol drug product, we refer you to the temporary guidance, repackaging and combining Propofol drug product during COVID-19 public health emergency, section 4, Appendix B. Thank you.
Thank you for responding to that question. We have one more question for Dr. Huang, and this is the question. What are the suitable container closures and storage conditions for the repackaged or combined Propofol drug products? Also, for repackaged or combined Propofol drug products with similar preservatives, what should the beyond use date be?
Thank you for the question. As you know, that the container closure system may affect the safety and efficacy of the drug product. So, especially for this stereo drug products, such as Propofol, they are suspected to microbial contamination and degradation. In addition, the drug substances may absorb to the container closure system, which may result in the efficacy issues. So the container closure system used for repackaging should be similar quality to the original containers and should be suitable for the repackaged product compatibility and maintain stability through the beyond use date. The storage conditions for repackaged and combined drug product should follow the requirement provided in the guidance. The recombined or repackaged compound, the drug product with similar preservatives, the beyond use date is not more than 4 hours. Thank you.
Thank you for responding to that question. The next group of questions will be directed to Dr. Feng. And here's the first question. For case number 2, how do we determine whether BE is deemed to fail or pass with 60% of the subjects recruited?
Yeah, thank you. For case 2, how to determine whether BE deemed to fail or pass 60% of subject. The applicant was planted 60 subjects, and then you can looking at the BE. For example, if the TR ratio of measurement is about like 60%, then you know that this formulation is deemed to fail, so that you can stop earlier to prevent to add additional subject into the study. But if it's not fail, like it's about 85%, but because the subject not sufficient, and you look at your CV percentage, and then you're looking at how it's possible to pass, then you continue the next stage. Thank you.
Thank you for responding to that question. We have the next questions also for Dr. Fang, and here's the question. What should be the study power of a clinical study if conducted in children?
Thank you. For the children, you're still using the same study power for the clinical study because those subject will still be the same. They're using the same statistical analysis and approach for your studies. But for the adaptive design, you still can use in the same statistical analysis for your data analysis. Thank you.
Thank you for responding to that question. We have one final question for Dr. Fang, and here's the question. Regarding adaptive design presentation, can you please elaborate what considers sufficient information and justification should be made prior to data lock and unblinding in slide 16?
Yeah, thank you. So regarding the justification and sufficient information should be provided. That's for the case during the COVID-19 pandemic, because 20 subjects was affected out of 60 total subjects. So that means 1/3 of subject was removed from the study. The study power will be a lot lower. So that means you can design your study, add another 20 subjects into your study, and pre-specify in your study, in your protocol, and then inform the agency that this study is because COVID-19 impact. So you can use a type of adaptive design to add more subject into your study, but you need to do it prior to data locking and unblinding. Thank you.
Thank you for responding to that question. The next group of questions will be directed to Dr. Witzman. And here's the first question. If a test product batch used in the conduct of our BE study expires while the study is interrupted due to COVID-19, can an additional test product batch be manufactured to resupply the study when the study resumes?
Good morning. This is a little bit longer of an answer than some of our previous answers, so everybody just bear with me, and hopefully I'll be able to give you a substantive answer here. So first, I think if it's a BE study and the test product has expired, you may use the test product beyond its currently established expiry date period, as long as you have sufficient evidence that can be provided to demonstrate that the test batch is still acceptable. So for example, you have storage and stability data beyond the current expiration that's in compliance with the finished drug product shelf life specifications, and you provide that within your ANDA submissions. And ultimately, the decision whether or not that information would be acceptable will be determined during the time of the ANDA submission and review. Another option would be you could provide information supporting a new test batch that would have to be the same formulation. It would have to be manufactured on the same equipment using the same manufacturing conditions. And and then if you had another situation where your test product expired after you had completed your in vitro bioequivalence study, your studies, but before you completed your in vivo PK bioequivalence study completion, you could consider your new unexpired test batches should be used in the in vivo PK/BE study, as well as repeating the in vitro BE studies on that new batch. And then if your in vivo comparative clinical endpoint bioequivalence study, for example, if that was part of what your recommendations were needed under the product-specific guidance for a product, if that were the condition, then the recommendations are to try to use the same test batch for all three of those studies whenever possible. And at least one of those batches should also be the batch that was used in the PK/BE study, as well as in in vitro BE, the in vitro BE data. And in those circumstances, we also suggest that instrument testing conditions should be the same as those used for any of the in vitro and or the in vivo BE studies that have already been conducted with previous test product and reference batches. And if you do have any differences in those, then any differences should be clearly indicated and scientifically justified within your ANDA. We do recognize that when you're doing a comparative clinical endpoint bioequivalence study, that those take longer, and those can sometimes, those are obviously the ones that would be most likely to be disrupted because they do take the longest time to conduct, and we recognize that that because of the COVID public health emergency, those could have been disrupted, and so we just recognize that if you do have a situation in which that has occurred, prior to submitting your ANDA, one of the best things that people can do is reach out to us via either a control correspondence or the pre-ANDA meeting pathway, either of those which may be available to you, and you can look at our guidances to figure that out, and contact us and ask us your specific questions ahead of time before submitting your ANDA to clarify those details, figure out which of those specific requirements you would need to meet, so that you can get your package in the best shape possible to submit your ANDA, so that you would be the least likely to submit your ANDA and find out there's a deficiency after the fact. And so that would be our recommendations for those situations. Thanks.
Thank you, Dr. Wismans, for responding to that question. We were experiencing a little bit of audio quality issue on when we were listening to you. So we're going to move on to our next panelist, and then we'll circle back around to you and ask you to just recap what you covered. The next question will be directed to Dr. Grosser. And here's the question. Do you see opportunities to advance and standardize new statistical approaches for BE evaluation out of the COVID-19 pandemic challenges? And if so, what are they?
Yeah, thank you. I guess I could follow up on something that Dr. Weitzman was saying, which is that there are a great number of statistical tools available that have been developed for the evaluation of new drugs. And we're seeing now the need for these in the development and the evaluation of bioequivalence of generic drugs because of the interruptions due to COVID. There are new problems in designs. So for example, I've seen, a case where there's a highly variable drug that has two studies going in, a fed and a fasted, and the interruptions, the lockdowns, occurred at different points in the studies. And so we have to be creative in using already developed statistical methods, but using information that we have perhaps from external sources in rigorous ways and in ways that we may not have needed to prior to these interruptions, but that will drive innovation in the statistical methods applied. I see us also using old methods in rigorous ways. So for example, when we're comparing a test product to 2 batches, you know, there are multiple ways of comparing, of testing these, adjusting for multiplicity, adjusting for the multiple comparisons, so that we don't use the standard 0.05, but rather a Bonferroni correction, a Hochberg or Holmes correction, all of which have been around in other areas of statistics for decades, but now we see a need for it in this time of COVID. So that's sort of where I see things going. You know, there are adaptive designs that, again, the mathematics has been worked out for the PK studies, but the clinical endpoint studies, the comparative clinical endpoint studies are another area where these methods can be applied, where development is needed, where, you know, the Office of Biostatistics here at FDA is working with OGD to further that development and to use these in practical ways to save resources, to rigorously determine whether products are bioequivalent. I saw there was a question for Dr. Huang about adaptive designs for irritation sensitization studies. So there, you know, where the best I can say to that, and maybe Dr. Huang has more to say, is that we're worrying about this, we're thinking about it, and we are considering these approaches in a variety of different drug products. Thank you.
Thank you for responding to that question. We'll ask another question to Dr. Grozer, and here's the question. Please clarify for determination of power whether less than 80% or greater than 80% to adjust alpha level at stage 1, what variability needs to be considered or expected based on literature or previous pilot study data?
Well, this is another case where, again, we're working on ways to use external information and literature, but and I'm not entirely sure I understand the question, but technically we can use the observed variance from the first stage to continue on past stage 1. And I don't know if that's answering the question. But that's the beauty of an adaptive design, is that we're gathering information as we go and we're adapting our knowledge, not only our statistical testing.
Thank you for responding to that question. We're going to check back in with Dr. Witzman, and we're going to repeat the question that we originally asked to allow her to give a recap on that. And here's the question: If a test product batch used in the conduct of a BE study expires while the study is interrupted due to COVID-19, can an additional test product batch be manufactured to resupply the study when the study resumes?
Hi, hopefully everyone can hear me a little bit better now. I apologize for the technical difficulties. My computer obviously thought it was smarter than I am, and it defaulted me to the wrong microphone. Rather than repeat the entire answer all over again, what I'd prefer to do for everyone, to spare those who could hear me, is to say that I would like to refer everyone to the guidance for industry, which is development of abbreviated new drug applications during the COVID-19 pandemic, questions and answers, which actually addresses this question specifically and talks about what to do at different stages when you need to, when your test product has expired and what the different options are at different stages during your development program. And if you have specific questions regarding your test product development, then you have options also to reach out to FDA. You can contact us via control correspondence, and if your product is eligible, you can also use the pre-ANDA communication pathway as well. So thank you.
Thank you for responding to that question. We have another question for Dr. Witzman, and here's the question. What if the reference drug is available under REMS only and needs to be administered at a qualified facility such as a hospital and access is severely restricted? Is there any other option for the use of the reference drug?
Thank you for the question. I think first, let me define what REMS is for those who may not know. A REMS is a risk evaluation and mitigation strategy, and those are specifically for products that have what we consider a higher safety risk to those products, and they usually come with something that's called an ITASU, or basically a step to evaluate and mitigate that risk or to assure safe use for the product. And those products in some cases can be, it can include additional training programs for the clinicians that dispense the medication or use the medication. It can sometimes mean that the medication has to be given only under certain circumstances, only in certain conditions. And for those products that have to be administered only in certain circumstances, like a hospital setting, there is little that we can do to change that kind of a setting. But I would refer to the guidance that Dr. Frost presented about for people to look at and use that reference specifically. There are clear recommendations that we've provided within that guidance document that can help people to figure out how to navigate and mitigate some of those risks for patients to be able to utilize those medications in that kind of a setting, to work with those hospital facilities to be able to still provide and run the studies that need to be run for those bioequivalence testing, while still being able to recruit subjects, be able to have your staff safe, your subjects, your study subjects safe, and to still work within the confines of a REMS program effectively. Thank you.
Thank you for responding to that question. And well, that's all the time we have for questions in our first Q&A panel. We'd like to thank all of our presenters and panelists for answering numerous questions that came in. We'll now transition into our break time and return at